Welcome to Flow Stars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry. Brought to you by Beckman Coulter at Bitesize Bio.
Today, Virginia Litwin, President-elect of ISAC, or maybe now, President of ISAC, joins me and she discusses the importance of diversity in science, setting up CYTO Women to advocate for equal opportunities, which has done so well within flow cytometry, and her passion for standardization to support clinical research. All this coming up in Flow Stars. Hi, I'm Peter O'Toole from the University of York, and welcome to this edition of Flow Stars. And today I'm joined by Virginia Litwin, who is President-elect of ISAC. How are you today, Virginia?
Je vais bien, oui. I moved to Quebec a few years ago, so randomly I had to learn French, and so randomly I will speak French. Sorry.
That's okay. Actually, ISAC was over, CYTO rather. So for those who aren't aware, CYTO is a big conference organised by ISAC. It is the big flow cytometry conference. Was over there not that long ago, wasn't it?
Yes, 2 years ago, completely independent of my being here.
Okay.
Oh, no, last year, 2023. Yes.
Yeah, Montreal, yeah.
Yes.
So, which was my first time to Montreal.
Oh, it's lovely, isn't it?
It was really good. It was really friendly, actually. I found it very welcoming. That was although it wasn't in their winter, and I've heard in their winter it could be quite cold.
Oh, it's lovely. It's lovely. We have about 2 feet of snow right now, and I debated about changing my background here to a picture. I live in the country, so of my snowy backyard, but I couldn't let go of my CYTO Women backdrop, which hopefully is still showing, yes?
It is absolutely showing. So it is interesting you chose the background of CYTO Women. So what for those who aren't aware, what is CYTO Women?
Great. Yeah, so CYTO Women is a committee within CYTO, or ISAC, and I was the first chair. So Johnny Moore, before she became president of ISAC, she put forth this idea to have a committee to address the unique challenges for women and opportunities and biases and things like that. And so CYTO Women was formed, and she asked me to be the first chair. And since this is informal, I will tell you my nephew's an artist, and so I hit him up to do the logo.
So I've never really appreciated the face in the logo.
Yes, it's to be, the idea was to be more ethnically ambiguous. And so when we formed the first CYTO Women committee, I picked a broad diversity of women, geographically, ethnically, age, so point in our career. And it was really, really fun to start.
This might be a difficult question. What would you say is the proudest achievement that CYTO Women has achieved to date? So that's a difficult question, isn't it? I would say while you're thinking about that, then that flow cytometry is probably one of the best represented male-female ratio of most of the technologies out in life sciences. I certainly in the UK, you go to them, the number of female attendees is really high as a proportion. The courses that we run at York, actually, unusually, is predominantly ladies, women that attend the courses over the men. That's not because women think that. It used to be the other way around, and I've noticed a big change in the last 20 years. How actually flow cytometry is a very welcoming environment, I think, for female and other diversities compared to many others.
I think so. I do have a friend here, my local BFF, who's an engineer, and that is still predominantly male, and maybe less welcoming, certainly. Yeah. And when I was in graduate school, there were my class, if you will. So we were immunology and virology, and most of us actually were women at that point. So
I've given you a bit of time to think about maybe the biggest achievement of CYTO Women.
I think it was getting it off the ground, really. And we had at CYTO 2021, I believe it was our second remote CYTO, we sponsored a viewing of the movie Picture Scientist. And if you haven't seen it, I totally recommend it. And I think it's available on Netflix now. So what we did there is we had to purchase the access to the film, and then people could log in if you went to the conference and view the film. And then we had discussion groups afterwards. We had, I don't know if you remember that conference, but we had lounges, right? We called them virtual lounges. And the discussions in there were really excellent. And in that, watching the film, I would have said going into it, oh, I've never experienced, any, challenges for being a woman in my career. And as I watched that, I was like, oh yeah. And so it just bringing that awareness. And I still have some colleagues, one friend in particular who's been a tremendous mentor to me, and he's older than myself, and just doesn't understand why we would have CYTO Women, and finds it insulting. And I think he's the only person that I've encountered that has said that. But I think opening up that dialogue of why we need to look at the point of view of other people, and why we need to be diverse. And I think for me personally, that is what CYTO Women has brought to me.
And any significant, I suppose that the individual you're talking about was one of the challenges, because you're being challenged by them for the remit. But did you meet any other obstacles along the way?
I think a little bit being taken seriously. I mean, you can't tell in the video, but I'm small, and so I think being taken seriously, treated as an adult. I have, yes, and of course, unwelcome physical advances type of thing. Yeah. And from big to small, right? From being told you didn't need to participate in a meeting, even though you were the subject matter expert at your company. So, and this was too recently to be acceptable, but being told you just needed to sit there and look pretty when you were the subject matter expert. So it's
Wow.
there.
And that was in an industrial setting, I presume, commercial setting.
Yes.
Yeah.
Yeah.
Wow.
So yes, and my first job out of college, it was a great lab. I mean, I learned so much from that lab head, but he later told me that someone else suggested he didn't hire me as a technician. He hired 3 technicians, and of the 3, I'm the one that remained in science, and so scientifically the most successful. But someone else at the university said, don't hire her. It's just a pretty distraction. So yeah, the things I don't think about, I don't, but watching that movie opened my eyes to that, or my recollections. And what I'm hoping is that did that for other people as well. Is like, maybe that is not the correct way to treat someone. Yeah.
It's hard to know where to go from that, because that's pretty shocking, isn't it?
Yeah. So
and I guess coming from a male background, I don't get to witness that side very often. So
yeah.
Anyway, so it's a very good reason to have CYTO Women, isn't it?
Yes. And I think also just that we had just the whole diversity for all groups. What do different backgrounds bring to the table? The way of thinking, what are obstacles that other people face? So just having that extra awareness has been tremendous. And keeping with this, I had the privilege to attend the Australasian Society meeting in 2022 or 3. I'm sorry. And we had a CYTO Women session at that, and we had some discussions, and we had a presenter for a Full Stop Australia, which was addressing sexual harassment. And we had then a panel discussion on that, and so much came out from the panelists, and then other even men in the audience were sharing stories, and it didn't stop at sexual harassment, just other obstacles that people faced. And afterwards, I mean, I was so drained, like I have never been after a talk. So I think that awareness has been the success for CYTO Women. Can we please talk about science?
Moving off the CYTO Women path, you said that you face some of this in an industrial setting. So where have you worked?
Okay. I started out, I knew I wanted to do biology, and I knew I wanted to do human biology. But beyond that, I had not found my niche when I graduated from university. And I ended up working with an immunologist, John Van Boxel, and he was just a great teacher. And so I just fell in love with immunology. And then we were doing microscopy with, I think you mentioned you had a microscope, fluorescent microscopy. Flow was still pretty new then. And then I ended up getting a job at UCLA, which was one of the leading groups in clinical cytometry, but it was just cytometry in those days.
Was this after your PhD at Iowa?
No, before.
Okay.
Yeah, so in the US, it's not that unusual to be a technician and work for a bit before going back. I know in Europe, you might go straight through. And so it was that group. I was the technician for a lot of postdocs. We were doing flow cytometry. It sounds like nothing at the time, but that was the group that asked the first question, because we're studying AIDS, what are CD4 levels in human T cells, or in humans? What are the level of CD4 T cells? And and that group became like my second family and supported me through graduate school. And they were, you should go to graduate school. And so they sort of pushed me forward to do that. And so I've used flow my whole career, and it's been a variety of virology or immunology studies.
What was your first flow cytometer?
It was, I think it was at UCLA. So I only stained the samples and brought them up. And it's embarrassing now because, I'm a leader in flow standardization and record keeping and all of that. But it actually, I left my samples, and yes, indeed, on a paper towel, I had wrote what I stained them with and left it in the flow lab. And later that paper towel came up in a meeting, and it was like, no, this is not acceptable. But so I think, I don't know, it was a single laser 3-color maybe at UCLA. They were working with Janice Georgie, was the lab head there for flow, and we worked with BD a lot collaboratively. So
okay, so probably would that be a caliber Eva at that point?
Oh no, not even a FAX4, because I think I don't remember. Yeah.
Wow. Okay, so what's the first cytometry that you used?
Well, again, in graduate school, I went to the flow core, and it was, but when I did my postdoc with Louis and at DNX, we had the benchtop model, which was, I think, the FAX4, what was it called? But we had a benchtop model.
Okay.
And then when I was at Bristol Myers, we bought a sorter, and I bought a Vantage. And then later I bought a Caliber, and then Kentos. So yeah. And I've just bought 4 A5SEs. So living up in the world.
That's a fair few. So you then went on to a PhD. Then what happened? So after your PhD, what did you do after your PhD? Was it straight into industry?
Oh, no. I went to DNAX, which I don't know if you're familiar with, but it was then a research institute owned by Sharon Plow, but completely independent. And DNAX is where Tim Mosman and Bob Hoffman worked, and Th1, Th2 concept came out of there. They cloned IL-10, they cloned GM-CSF, identified IL-10. So it was just a great collaborative place to do research. We had immunology section and molecular. And so we were funded by Sharing Plow, so we weren't having to do grants, and it was not at all competitive or cutthroat. And I worked in Louis Lanier's lab, who will be our keynote, our hook lecturer at CYTO 2025. And he was studying NK cells, which is, I worked on NK cells when I was a technician at UCLA, and also FC receptors. My graduate work focused on FC receptors. So it was, I did research there, and in the Bay Area, it was a lot of us went from there into biotechs more than into academia.
Yeah.
So I went to a biotech, and then to Bristol Myers, and then to a contract research organization where I was then, that's when I quit using flow cytometry as a research tool. And it was now, I got more focused into the technology, and I had to learn standardization and validation of flow cytometry.
Which, if I'm honest, it's got to be one of the, for me, one of the least interesting aspects of a role is that standardization. And yet, I presume you have a real passion for standardization.
I do, and it is not at all the least interesting. So I gave a talk recently, and talking about, how do you do rare events? I bet you think rare events are interesting, right?
I'm with you. Come on.
Okay, so it's everything you've ever heard at a CYTO meeting goes into a rare event assay. You need to start with a fully optimized assay, how we should optimize it today, not how we did in the last century, where you are really optimizing your signal. You are going to be able to identify dim events and rare events. And then you need to make sure you have the proper gating and the proper fluorophores. And then you need to understand what's the variation of my assay, and how low can how few events can I reproducibly measure? And how do I know that I have only those events in my gate and not everything else? Bad comp, bad doublets, bad spread. So no, standardization is everything put together.
And fundamentally vitally important, especially with the contract research agencies that you're working for.
Mm-hmm.
My goodness, they have to be on the ball and near perfect.
And so when I started working at Bristol Myers, I was doing the phase 1 clinical trials, and that was, I think a lot of people thought, oh gosh, working at a pharma would be like not fun for a scientist, but it was really exciting because I was doing the biomarker assays for the first in-man trials, mostly for oncology, some immunological diseases as well. And it was really exciting to see the patients respond. What new target are we hitting, and how can I support that with my biomarker assay? So
it really is. It's very close to front line, isn't it? Because you're getting very close to direct impact.
Yeah, yes. And then it's so for people that aren't here, I latched on to the idea of, translational science, translational medicine. So if you're starting in a research lab, and in fact, the things I did in my postdoc are now going into therapies. So you want to make sure that your data is as robust as possible, so that if someone takes it to that next phase in translational science, that it's repeatable and it can move on.
So within those roles, you're obviously a big advocate for not being in the academic world, that there's a great role outside the academic roles and environment. What would you say the biggest advantages being working outside of academia?
I don't know. With my role at ISAC, my new role, I'm the first ISAC president that's not from academia. So I do interact with a lot of academics. I don't know. I think probably for me, it's having that impact. So can I have an impact on the program? Can I help my clients do a better assay? Can I help that drug? A lot of people say, oh, we're in a CRO, you know, our goal is the patient. And for me, it's the drug. I want that drug to go forward, and I want to, if it should, and I want to support it so that the regulatory agencies can make the right decision about that drug, which ultimately will help people. So for me, it's that impact, but it's not to say you don't have it in academia. So
does CYTO actually have a section or a meeting for commercial flow cytometrists?
we have, I think I did my first workshop at CYTO in 2013. And I think a lot of people were surprised that, yes, this is interesting. Or people in pharma and CROs are doing interesting work. And so we've had workshops almost every year since. We do have a flow group within the American Association of Pharmaceutical Scientists. But I think what we're doing, we're well represented at ISAC, and the standardization, the validation, we're bringing that forward, but it doesn't need to be its separate group.
Yeah. It was just thought, so the Royal Microscopical Society, we don't have anything for that side of it, although we've got obviously industrial people on the different sections. But there's a movement in the UK called LRIG, which is which is not for profit, but it's commercially orientated for people in pharma, biotech, standards, and brings in academia as well. So it's sort of hybridized them, but has a very much focus on that biopharma side. So I just didn't know if there's anything equivalent in the States, or whether CYTO could look at that across the pond.
LRIG, yeah, possibly.
It's just a thought while we're talking. On another side, so actually, I obviously, this is very organic. I did have to. I've never met you before in person, so this is amazing. But what I was blown away by is just how many different societies that you're involved with as well. So obviously, CYTO in a very ISAC in a very big way, but also the APPS, so the American Association of Pharmaceutical Scientists, ICCS as well on there. So you're on the advocacy committee for that. You're on the community section for every. Why so many societies? Why active on so many societies?
So going back to my first job in pharma, where our lab head was forward thinking enough to bring in my role, and so I was doing the cell and molecular biomarker assays, and he wanted to bring up flow cytometry, and then really he was paralyzed by the flexibility of the technology. And that's where, not really in the nicest, easiest way, but I was sort of pushed in the deep end of the pool to learn standardization. And I went to my first ISAC and I was complaining, and Joanne Lanigan said, Virginia, he's right. And ISAC had just, well, there was no clinical cytometry then. So they hadn't split. So there was a big movement on standardization. And so I don't know. I guess what you're saying is I needed to pull, when you're doing biomarkers, you're not clinical and you're not research. So you're pulling from both. And then friend from UCLA, John Ferguson, I started a group within AAPS, the flow cytometry group, because we thought, okay, we want to push the industry to talk about how should we validate flow cytometry methods, and how can we use this technology to support drug development. And so we picked AEPS because we thought they were more aligned with the regulatory agency. And then at one point, someone at the FDA told me, no, just do a clinical standards guidance document. Just take that information after you've written all those papers, and maybe then we'll pay attention to you. So it's sort of, I guess, we're chimeric here. We need a little bit of each.
So what's your favorite society?
Well, ISAC now.
It was a loaded question, wasn't it? So, how are you looking forward to becoming president?
I think I've settled in the role. At first, I was just a little bit. It was a little bit too daunting. You know, we're vocal. We have a lot of brilliant people, and so I was a little bit overwhelmed. But now I think I realize what my skill set is that will be of a benefit to the society, and so yeah, I'm really looking forward to it, and that is to some of the past presidents talk to me pretty often, and really passionate about keeping the society moving forward in all our diversity, right? The engineers, the programmers, the SRL, the researchers, the pharma people, and so I just want to make sure we have a big tent and everyone benefits and the society and the technology grows.
So, what do you see as the biggest challenge for ISAC going forward? And
We're, I think we're at a good place now. We went a few years ago from being a managed society, and now we're have independent management, and that's been, nothing is as straight exponential curve there. So I think as we continue to grow and bring in new members and bring in diverse members, right? So I think that's our challenge. I think we'll do it for sure. But that's it, is bringing in younger people, getting really good scientific diversity in our tent.
I probably going to change subject completely now. You heard how you got into science. You wanted to do human biology with your first degree, which I think was Santa Barbara. If I take you back to when you were 6, 7, 8, 9, 10, those very early days, can you remember what the first job was that you ever wanted to do? What you wanted to be?
Probably a ballerina, but I really wasn't very good.
It's a good answer, though. From a ballerina, was it straight to a scientist? Was there any intermediate steps you're thinking, oh, I quite fancy that job?
No, I think that was it. I have more talent in the science world, and I think for me also it was very obvious for me that's where my aptitude was. And much as I love ballet, I wasn't really the best, So
That's a good answer. And you're still dancing through your career, so arguably it's not so far off it. It's just in a different way. If you could do any job for a day or a week, a day in the life of another job, not in clinical or academia, what job would you like to sample? What would you like to try?
I've never thought of that.
You never looked at someone who's working and think, oh, I'd love to know what it's like to work alongside them or to do that job, or
No, it becomes obvious to me jobs I couldn't do. Like I write a lot, but like journalists I'm fairly good writer, I think, but I mean, I am slow. Every word is just a pain. It's a slow birth. And journalists, I think, what they have to come up with that on the spot and go into war zones and write a good story off their cell phone. So I think of jobs I can't do, but I like to cook. So maybe my husband always tells me I should be opening a little restaurant, which again, I'd be terrible at, but I do come up with some nice menus for my N of one customer here.
so actually, I was going to later on. I'll ask some quick fire questions, but I'm going to bring one of those forward now. I usually ask, what is your signature dish then?
Know that I have one. I'm vegetarian, and I've been vegetarian since I had my first apartment at university. So what I like to do is put together a full menu. So I just as always take on too much, and so I'm just going crazy at the end. But I pull it off. So and I'll tell you this: during COVID, we of course couldn't go out to dinner, and so we started having dinners at home on the weekends so I could spend more time cooking. And and I had to learn French to get residency here in Quebec. So I started posting on Facebook every week my menu in French, and I called it Cafe Corona. So I had a lot of nice dishes there. It's just vegetarian cooking.
And you published those online, did you say?
On Facebook till we all got too sick of it. Yeah.
And did your friends and family who looked at it, were they trying it, commenting, feeding back?
Oh yeah. Oh yeah. Yeah. So I mean, for years I've posted some things. Yeah.
But you were posting it in French. Can all your family do French?
You know, and my attitude now was I have to learn French, and the rest of the world is going to suffer with me. If I feel the need to express it in French, I will. Don't put me on the spot here, please.
How have you found learning French?
It was good. It's harder being somewhere that's bilingual, and so the Canadians being so nice, they hear your accents imperfect, and so they might answer you in English, which I take as a huge insult. That meant I really was doing poorly. So it'd be easier if I was in France for sure.
Okay.
So I you know, this is going out there. Anyone wants me to do a 3-month stint, talk at a French meeting, I'm there.
Why the move to Canada?
I came here for a job, and I had a couple of opportunities at it was in 2017 I moved, and you know, we sort of decided as a family. I said to my husband, I said, well, pick one of these places. Where do you want to live? And he said, well, I can't pick, it's your career. And I said, I'm not going to give you anything I wouldn't want to take. So yeah, we came up to Montreal.
And how's his French coming on?
He had more French in school than I did, but only one person in the household had to take the French exam. So he has been extremely lazy about it, yes.
And you have a pet cat, correct?
Yes. And Pierre.
How's his French?
Pierre is completely bilingual. He speaks French avec moi et anglais avec Richard.
So, perfect. Oh, it's a really bad pun. Sorry for the pun on the perfect bit, but there you are.
Yes.
As it works, so how have you found relocating? How have you found that cultural change? It's always a bit out of comfort zone moving to a different city, let alone a different country. So how have you found all that?
Right. We've moved a lot, but yeah, it was more challenging here, and having to go through immigration and everything. And I'm American. I grew up in the US. So yeah, being an immigrant was really it was a lot, but I finally got permanent residency. It's a lot of forms and a lot of money and a lot of everything. So I had no idea how that was, what that experience would be like being an immigrant. But and even though there's you know, relationships between the US and Canada, especially for certain positions such as biologists, that you're a preferred profession. So I had it easy in that regard. Still, it wasn't all that fun. And I had just started a new job, and I had just started the kickoff for CLSI H62. So all of that, trying to learn French, and full-time job, writing an international guidance document, there wasn't a lot of time for that.
Which I guess means you kind of just moved and didn't worry about it because you had so much going on.
Yeah, it's like, yeah, you just I never think through all the details, whereas other people will really look. I've asked other people to, you know, take over committees for me or this or that, and they're like, how much work will it be? Will I this? Will I that? And it's like, I good or bad, I never ask myself those questions. Sure, I'll do it.
So you've got the big document done. You've done you've got the presidency of ISAC. You've been on the Clinical Lab Standards Institute as an expert panel member. You're actually an associate editor for Cytometry Part B. Have you ever stepped back and appreciated how much influence and impact you've had on the field?
Do I step back and think that?
Have you ever just stood
No.
back and think, actually, I have actually contributed quite significantly. I've done quite a bit. Have you ever actually just taken time to reflect on your own impact? It's common for us to look at others. So I'm looking at you now thinking, my God, you have done so much. You've done so many, and really quite important impacts as well. Not just with ISAC, but actually in your professional career, not just in your charitable societal career. Have you ever taken that time and stepped back to appreciate what you've done?
No. I I don't think I didn't get that gene. It would just seem weird and arrogant. I no. I mean, I'm happy when I see the guidance document or papers that I've written referenced by people I don't know. But I didn't, no, not quite like,
So the Wallace Culture Award was it last year or the year before?
yeah, last year, yeah.
Recognition of your contributions to cytometry in the field. Surely then you must realize that actually you have had you should be proud of what you've done.
Well, it was very nice, and I'll tell you, a lot of my people that were in my labs were there, and my mentors when I gave the talk, and it was really nice. But no, I could never just sit around and say, oh.
No, I guess you're not a vain person going to be doing that, but it
Well,
must be nice to receive that and just think, oh, actually, gosh, without realizing it, you've done this because it's just what you do. It's what your passion has been, but it helps the community.
well, I think more than that is I feel privileged, right? So and when I got the Culture Award, it was a career retrospective talk, but I talked about my mentors and how they helped me. And then the end, it was like, this is what we need to do. And ISAC and ICCS as well, very focused on mentoring that next generation of scientists. And with my role at ISAC I feel privileged to be in the room, to be talking with people like you, working with Jessica Houston, Johnny Moore, all the Pauls, Robinson, Wallace, Smith. So yes, it is a privilege, and I am really grateful. Yeah.
So who you mentioned your mentors, who have been your biggest inspirations?
When I started at UCLA as a technician, there were a lot of women, right? And they some are still my friends. And Janice Georgie, there's now she passed away from uterine cancer, and there's now a Young Investigator Award at ICCS for Janice. And so they have really been a lot of those people my mentors. But I mentioned them in the culture talk. But Susan Plager was a new postdoc at UCLA, and I was her technician, and she was, now we call it sponsorship, but she recommended me for my first anything committee, and continued to support me. She helped me with the qualifying exams in graduate school. She put me on my first NIH grant, my first editor role, and I don't know why. And Johnny Moore was also very instrumental in recommending me for things. And I think without them, I just had these great opportunities to do these things. And so those are, I don't know, inspirations, but they were just so enablers for me.
So that makes, just thinking about it, of helping your mentors, your inspirations, and how they're stepping you up. Some of that would have been delegation. And how would you say you manage delegation? Are you a good delegator?
Terrible. And I still, have to muster up, some courage, and, to ask someone to do things, So yeah, that is not something I am great at, So probably I spend more time doing things I shouldn't be doing, and yeah, but
Okay, so some quick fire questions.
Okay. Quick
Are you an
fire.
early bird? Quick fire. So here we are.
Okay.
Are you an
Okay.
early bird or night owl?
Early.
Early bird?
Up.
PC or Mac?
Oh, Mac.
Mac. McDonald's or Burger King?
Neither. I'm vegetarian.
Yeah, I better say that's not going to help, is it?
Although my first paying job was McDonald's.
Okay. Uh, okay. What's your favorite takeaway food or your favorite fast food?
Starbucks
Okay.
coffee with almond milk.
My son works at Starbucks. That's good. Chocolate or cheese?
Oh gosh. Chocolate.
Ah.
Although cheese and port is good too.
Did I say coffee or tea?
Nope. Coffee.
Coffee. So chocolate, coffee go well together. Beer or
Mhm.
wine?
Wine. I'm from California.
Right.
White or red? Was that it?
Yep.
Oh, well, I better say red if Paul Robinson is listening, but I like them both. Yeah.
Okay.
He'll divorce me if I say red or white. So,
What is your ultimate favorite food?
ultimate favorite food. I think on Sundays we go, well, we used to have the paper, now we don't, but we would go for a special coffee and a pastry somewhere. So I would say right now it's a superb almond croissant, but maybe not an ordinary. And we have some great bakeries here in Vermont.
Okay.
So yeah.
Whilst it's warm or cold?
Either, that's fine.
Okay.
Yeah.
What is your food nightmare other than meat?
Um,
Any style of food or
oh,
thing?
too spicy, too spicy. I am a wimp for spices, so
Yeah.
I can handle a tiny bit,
Do you prefer to cook or clean?
Oh, I love them both. I love cleaning. It's the OCD, which is great. I have a virology background, so it's perfect.
Oh, after cooking? You want to clean up after cooking?
Yes, yes.
Oh, your husband's very lucky.
And well, for so many reasons.
TV or book?
Book.
What are you reading at the moment? What's your favorite genre?
Um, it does just some good writing. Not scary. I've done a lot of biographies. I just love David McClellan. Um, yeah, not trashy, fluffy, but good writing.
Okay. Favorite film?
Favorite film. Oh, I don't know if I have a favorite, but I do like a good movie.
Oh,
Just haphazardly, we watched on Netflix recently Bridget Jones' Diary. I was laughing from the get-go, from the credits. It was funny. So I think a good, clever movie.
okay. Star Wars or Star Trek?
Neither.
And favorite color?
Brilliant Violet.
Oh, so few guests ever come out with something like Brilliant Violet or Fluorescein or FICO Erythrin. Most go blue or green or yellow. It's like, come on. Well done. Proper flow cytometrist. At last, you passed the test. Uh,
Yes,
what's your favorite music? What do you like listening to?
I like I like rock. My husband's a bit of a rock historian, I would say. So my rock, and I'm the youngest of a large family, so I think my music tastes are above my age. But yeah, I think all of the early good rock.
Okay.
A lot of the women singers, Joni Mitchell, Emmylou Harris, Linda Ronstadt, all of them.
Okay. So that's good to chill out to. Do
Yeah,
you
yeah.
have any hobbies? So if you've had a stressful day at work or in your life, what sort of hobbies do you do to chill out?
Um, well, I pet Pierre, and I put my head on his little chest and listen to him purr, and it is better than heroin, although I've never really tried heroin. But that's very good. I'm a runner, So, yep, I put some music on my Sirius radio on my iPhone, and great running here. Lots of trails.
What's your distance?
An hour,
10K-ish?
10K-ish. Yep, yep. I'm not fast, but I don't run
Miles?
for
I should convert that for the Americans, shouldn't I? 6 miles?
yeah. Yeah, not terribly fast, but I'm just going for sanity and fun.
Do you do any events?
I do. I did one recently. My local BFF, the engineer, is she used to do marathons, and so I'm such great company. She suffers through my slow, shorter runs, and we did a 10K a while ago, and I want to do one this year as well.
But she hasn't persuaded you to do a marathon yet with her.
No, she wanted me to do a half marathon with her, and I wouldn't. So I'm
Why
fine
not?
with. I'm, my feet are
Do it
happy,
just
my
once.
knees are happy. Yeah.
Yeah. So once you've done the half marathon, you know you're halfway to a marathon, then you have to do the marathon. So maybe you're better not doing the half because then you're not.
I got enough goals, right? I'm doing another CLSI document, CYTO 2025, blah, blah, blah. Yeah.
Okay. So outside of those hobbies, what would you say has been the most challenging time of your career where you probably had to lean on those more often?
I don't want to share that.
That's cool with me. So I'm gonna flick it round and say, what's been the best time of your career? If you could ever live an era, a year, or a moment in your career, what moment would you go back and relive?
It's right now. I mean, last year was the even I did the ISAC presidency, and before they had the special election, Paul Robbins had approached me about that, and I'm like, Paul, it's a year off, like and that took a long time to think about, could I do it, would I want to, and then longer to run it by my husband, what would you think? And then shortly after that, the Coulter award came up, and I had been nominated before, and no one walks around thinking, oh, I hope one day I'll get the Coulter award. But when you don't get it, you're like, because people tell you, oh, I nominated you, and you're like, yeah, I wish you hadn't shared. But so it was just really special. And right now, it's with my role at ISAC and with the conference, it's like, okay, what can I do to shape something meaningful to really make a contribution to the society and all? And a lot of my people that I've mentored or have been in my lab are doing great, and I'm so proud. And they give you a drop of credit, and it's so I would say right now it's just nice.
Okay. And do you have any future predictions of where we should be going flow cytometry wise?
Yes, we need to be quantitative. Absolutely. We need to be, yes, I'm working on that now. We need industry behind us so that the software will work when we quantitate our data, and then our variability between everything will diminish, and we can move forward, and we can use those data in that whole translational space.
With all the flitting, moving in a different direction again, with all the different standardization and standards, do you do much with NEQAS?
I did when I was at clinical cytometry, because it's the equivalent in the US would be College of American Pathologists, and I know not what I'm doing now so much, but yes, when I was more involved with ICCS, clinical cytometry.
I only asked, I'm good friends with Liam Whitby.
Yes, I know Liam.
Okay.
And I knew David Barnett was a contributor to the guidance document as well. He was on that.
So I think Liam took over from Dave, didn't he, when it came to NEQAS as president and CEO. And actually but I think he was on the first series of the Flow Stars.
Oh, well, yes.
Brilliant. So there you go. There's one to go and watch. Although I would say we did it in an evening or at dusk. and he got darker and darker and darker. So actually, if you listen to it, it's fine. If you watch it, you might as well just listen to it. 'Cause at the end, he goes very dark. He's also a runner, isn't he? So he also has that in common. So he's
Yes, yes.
passionate about standards. So passionate. So again, similar to yourself, so
Hmm.
passionate about standards and the environment and supporting. So yeah, I guess a UK equivalent almost to
Yeah.
yourself in many ways.
And they've done a lot of really good papers and really good work on saying, okay, we put out the standard, how does it really work? Let's do a interlaboratory study on it. Yeah.
Because I've had Dave in York, but Liam as well, and others from NECRAS and standards, because we run the RMS flow cytometry course, which has a whole clinical session. So, for 3 days of clinical. So there's a lot of content around that with different tutors coming in, of which Liam, it's always good to see Liam. He also likes coffee, like yourself, and chocolate. A lot of similarities.
Chances.
How much throughout your career, how much have you managed to travel?
A lot. I mean, less than some, but a lot. Yeah. That's great. I just did my first live ed course and in Buenos Aires, and it's just fantastic. And I work from home now. I oversee actually 6 different laboratories, but 3 different business units, and which is fine. But I had forgotten how much fun it was to be in a lab and around the students, and oh, it was just great. Yeah.
So would you say Buenos Aires is your favorite place that you've been, or?
It was pretty nice. It was pretty nice. No, and I was supposed to go to us, New Zealand, but I didn't realize they were both so far away, so I had to postpone New Zealand. I know I would say Australia. I do have a soft spot in my heart for Australia.
Any particular part?
No, let's see, where have I been? No, it's just great. And my husband and I are real animal lovers, and we love wildlife, and it's like, wow, look at these animals. So yeah, it was great.
Yeah, because the wildlife again is so different. The bird life there's quite a lot of endemic. It's yeah,
Mhm.
very good
So
for birding and whale watching.
yes, yes,
So
yeah.
what's the birding like? I should know this off Claire Brown, who's in Montreal. So she's quite a good birder. What's the bird life like in Montreal?
It's like New England. My husband's from New England, so it's not different, right? When you cross the border. But very nice. Cardinals, blue jays, those are our flashiest birds, are very, very pretty. Nice goldfinches in the summer. The occasional, if you're really sharp, a scarlet tanager.
Yeah.
Yeah. Yep.
Ah,
Of course, your robins, juncos, chickadees. Yeah.
bizarrely, I think it was the first scarlet tanager that was actually in mainland UK. Was twitchable. So it actually was about 40 miles away from where I live recently.
Wow. Wow.
And I didn't get time to go and see it, but there you are.
There you go. Woodpeckers. Yeah.
So I think we are probably up to our time as my watch just beeps. I have to ask, is there any last thoughts or any top tips or advice for someone starting out in their career? What would you advise them?
Yeah. So I think what like I look at my career and I think I just fell into it, or my career found me. And I just think I was very lucky. And people say, oh, you did the work, you shouldn't say that. But I had good opportunities and I had good mentors. And so I think you just have to follow the road, right? And I think if you're not happy somewhere, just leave. And you will find a good mentor. And I think just exploring where, lean more and more towards what you're good at and where you can contribute. Because I mean, I never, especially, with being, the incredible group we have at ISAC, I never feel like the smartest one in the room, but I know I'm making a contribution and I'll do the work. And so I think that's just it, is just do what you're good at, do what you enjoy, right? I mean, I wanted to be a ballerina, but I was not good at it. So
How many hours a day do you work?
I don't count. We don't ask that question.
Yeah, I've got a feeling I know the answer. Virginia Litwin, thank you so much for joining me today. Thank you everyone who's watched or listened to the podcast. Please do go. You've heard Virginia talk about Jessica Houston, Johnny Moore, Paul Wallace, Paul Robinson. We talked about Leah Whitby, all previous guests, all equally inspirational, all with their big impacts as well. And Virginia, you should look back at yourself at some point and reflect on just how much you've contributed to the whole community and to science in general. Virginia, thank you for joining me today.
Thank you.